Yes, she records.
Yeah, that's what I understood, yes.
Yeah, she records now. Okay, so I can start?
Yes, go ahead.
Yeah, okay.
Yeah, great.
So thank you for the opportunity to give this talk. Today I will speak about the GMO-free RNAi and particularly about double-strand RNA pesticides, which is a major part of the project duration that we are running now with Dimitrios and other co-workers. First, for those who are not very familiar, I will introduce RNA interference or RNAi. RNAi is a manner for RNA-mediated gene regulation. It's triggered by double-strand RNAs, which are processed to small RNAs of 21 till 24 nucleotides, and they can trigger mRNA degradation or protein translational arrest or DNA methylation. And RNAi is conserved among eukaryotes. And for their discovery, Craig and Mello were awarded the Nobel Prize of 2006. So let's focus on plants for the moment. So the RNAi or small RNA, the siRNA, which stands for short interfering RNAs, in plants this is the pathway. First, the double-strand RNA is processed by endonuclease called dicer4 or DCL4 into 21 nucleotide small RNAs. And one strand of these small RNAs will be loaded on a protein called Argonaut or AGO1. And then by base complementarity, it will recognize complementary mRNA in the cytoplasm and cleave it. That's why we have the RNA degradation. But besides dicer4, another enzyme, dicer2, competes with dicer4 for the processing of the double-strand RNA. And processes the double-strand RNA to 22 nucleotide small RNAs, which are loaded again in AGO1. But this time, besides cleaving the target mRNA, they also recruit an enzyme called RNA-dependent RNA polymerase 6, the 3' of the target. This is important for the next slides to remember. This RDR6 enzyme. And now this RDR6 transcribes from the very 3' part, the 3' part of the target into additional double-strand RNAs, which are again processed by dicer into small RNAs, leading to the generation of secondary siRNAs. Thus, with the RDR6 enzyme, the RNAi phenomenon in plants is getting into a self-amplification reinforcing loop. Okay, so we have amplification of RNAi with RDR6 enzyme. And then finally, another enzyme called dicer3 or DCL3 will compete with the other enzymes for processing of the double-strand RNA and cleaves the double-strand RNA into 24 nucleotide small RNAs. Now, these 24 nucleotide small RNAs get loaded onto a protein called Argonaut 4 or AGO4, and this complex of 24 nucleotides with AGO4 goes to the nucleus and recognizes either the complementary DNA or a nascent transcript produced by polymerase 4 and methylates the DNA. So that means, to cut the long story short, plants with a single double-strand RNA, we may have either RNA degradation or transitivity with RNA degradation or DNA methylation. All of these processes are possible. Now let's go for the GMO-based RNA. This is because of this potential of this RNA as a tool. Line biotechnologies, of course, get used to it. And they generated transgenes expressing double-strand RNA, inverted repeat transits actually, that upon transcription, they have the potency to form into a double-strand RNA and trigger RNAi. So why they did it? They did this in order for various purposes. One of these was to silence some endogenous genes, as you see in this example. You have these apples and these potatoes that express a double-strand RNA against polyphenol oxidase. Thus, these plants, transgenic plants, are resistant to browning. And then you may also have some tomatoes, transgenic tomatoes expressing a double-strand RNA against ACC synthase, and thus these these fruits are resistant to premature ripening. And of course, also there are some transgenic maize, corn plants expressing double-strand RNAs against insect genes. And these plants are resistant to Diabrotica virgifera. These corn plants are actually commercially available as well. They are the Smart Stacks Pro. And in another very interesting example, transgenic papaya plants, you see huge plants here, are expressing double-strand RNA against the virus, papaya ring spot virus. And as you can see, it's very impressive, this picture, that they are very resistant to the virus. Okay, so unfortunately, this potential of genetics, this great potential of genetics to improve agriculture is not appreciated by everybody. And perhaps because it's natural to be afraid of what we don't understand. The biggest part of the public is very hostile against GMOs. And of course, there are some reasons to there are some sound reasons for this, but in my opinion, at least, most of their arguments are really groundless. But in any case, the fact is, things are how they are. And now, with the exception of some parts of Asia and US, in Europe at least, GMO plants cannot be grown, but of course, GM products can be imported. But in any case, it seems that the future of GM plants in Europe is not very nice. Let's see if this will change. But since we live now and not in the future, that things perhaps may change, we have to address the issues now with the current policy and the current public opinion. So we also, as biotechnologists, we try to avoid GM plants, GMO organisms, and but in order to take into advantage the RNAi as a tool, what we do now, we do not produce plants expressing transits that culminate in the generation of double-strand RNAs, but deliver directly the double-strand RNAs to plants. Okay, so this is the GMO-free approach. You don't have a transgene that is potentially expressing the double-strand RNA, but directly apply the double-strand RNA or the small RNAs to plants. So while in the first case with the GMO approach, it's called, you may find the literature called the Higgs or host-induced gene silencing, because the host expresses these double-strand RNAs. This GMO-free approach, you may also read it as SIGS, spraying-induced gene silencing, just to stress the fact that these double-strand RNAs are not expressed by the plant, but they are sprayed, they are exogenously applied. So, of course, the one issue is whether these double-strand RNAs that have the potential to be used as pesticides are safe. And there are two very helpful and important documents from OECD, one from 2020 and the other from 2023. The first one from 2020 has made an extensive survey of the literature and also, let's say, decided or suggested that exogenous double-strand RNAs are safe for the environment because they are rapidly degraded in the environmental conditions, and they have very minimal potential to induce RNAi or other undesired effects to non-target organisms. And just this year, another document from OECD addressed the issue of the health, of the human health, the double-strand RNAs in the human health. And and they also decided that, or suggested that the exogenous double-strand RNA is safe for human health, even if it enters the trophic chain. So personally, I find these documents convincing, although they are not conclusive. The current data, the current scientific evidence suggests that there is no visible threat of double-strand RNAs to the environment or the humans. But of course, further experiments, further assays need to be performed to be sure about that. Okay, so if we conclude, or if we agree that the double-strand RNA as pesticides are safe and can be used and can reach the market, one issue is how to generate these double-strand RNAs, at least in the lab in the first case, just to see if they are suitable for us, if they can be used as herbicides or as pesticides or insecticides or fungicides, etc. So if you, the most easy way is to select, of course, your gene of interest, whether this is a plant gene or a viral gene or an insect or a fungal gene, and then you don't need to have the full gene, the full length of this gene. Just a small part of this gene is fine. So you clone this gene into a plasmid that has two convergent T7 promoters, and that upon transcription, double-strand RNA will be generated. And then you make RNA extraction from this bacteria, and then you can apply your double-strand RNA. The advantage of this method is that it's quite cheap, because it has been estimated that it's approximately €1 per gram of double-strand RNA. This is really cheap. But the big disadvantage is that these double-strand RNA that you extract from the bacteria are not pure, because they will contain, besides the desired double-strand RNA that you want, all the RNA that is present in the bacteria at that moment. And moreover, and this is most important in my opinion, is that you may also have a cross-contamination of bacterial, of transgenic bacterial in your RNA preparation. And even though this contamination may be minimal, it's important and it should not be neglected. So in order to avoid this contamination issue, with possible contamination issues with GM organisms, another method, which is even easier, is to select your gene of interest and make a PCR with primers covering, let's say, 500 base pair of your gene of interest, and with primers having T7 promoter tails. This T7 promoter is it's a minimal promoter. It's almost 20 nucleotides, as you see here. It's a very small sequence that the polymerase can recognize and transcribe the fragment, the amplicon here. So in that case, you avoid the use of bacteria and cloning steps, and you have a very pure and clean double-strand RNA, and with completely avoiding the risk of having some GM bacteria cross-contamination issues. So the benefit, of course, is that it's very pure, but it can be very expensive. For example, there are several available kits in the market right now, and at least in our hands, the best kits cost around €1,000 per gram of double-strand RNA. So it's very expensive, actually. But if you are not familiar with molecular biology procedures, or if you're lazy to generate this double-strand RNA and you have a lot of money, you can simply buy it. And there are several companies generating the double-strand RNA for you. These are some examples here. I'm not working for a company, just some examples that are available. One is the Genolution, the Korean Genolution that we cooperate with in a ration program. The other is Greenlight Biosciences, located in the US, and another is in Euphoria Biotech in Germany. So these companies they have a patented in vitro system to generate double-strand RNA. So they don't resort in bacteria. It's GM certified, GM-free RNA. And the price for depending on this RNA, depending on how much you order and the company, ranges from €1 to €10 per gram of double-strand RNA. So, but it depends. This depends, and this is not very expensive if one considers that the double-strand RNA pesticides may reach the market or the field, because the application dose, especially for insects or for viruses, is approximately 10 grams of double-strand RNA per hectare. That means that one would need approximately €10 per hectare of the cost of double-strand RNA pesticide would be €10 per hectare, which is not a lot. Of course, you may need several repetitions of the spraying, 2 or 3 repetitions, but even the cost of 50 or 30 or 50 euros per hectare is not very prohibiting. Okay, so some critical issues to consider when designing the double-stranded pesticides is the first is the RNAi trigger, meaning the double-stranded RNAs or the small RNAs. When one chooses double-stranded RNA as a pesticide, then the length of this double-stranded RNA should be approximately 200 till 800 base pair. It should not be less and it should not be more. It should not be less than 200 because the dicer enzyme, which recognizes double-stranded RNA and cleaves it into small RNAs, is not easily recognizing double-stranded RNAs with a small length. For example, a 100 base pair double-stranded RNA will be inefficiently processed to small RNAs by dicer. Okay, so you need a length of approximately at least 100 or 200 base pair. Okay, this is the minimum size that you need. And then, of course, you should not exceed this length to 10 kb, for example, because the larger the RNA, the less efficient it anneals into double-stranded RNA. So an average or a golden standard for efficient double-stranded RNA is 500 base pair. I would say 500 base pair would work in most cases. Of course, one issue to take into consideration is that with the double-stranded RNA, it will be processed by dicer into several small RNAs, a plethora or a diverse population of small RNAs, some of which small RNAs may recognize other targets, other transcripts, and cleave them. Thus, you have the off-target effects, which is completely undesirable when designing pesticide. In order to avoid this risk, one may resort to application of small RNAs, of discrete small RNAs of 20 or 22 nucleotides small of size, by which the risk of off-target possibility is significantly minimized, and it can even be excluded if one has access to bioinformatic to databases and to check whether the small RNA, this 20 base pair, will have a potential target on a non-target organism. And the second issue when designing a double-stranded RNA pesticide is the target. Which target should we choose? The target, potentially any gene can be target. For example, in insects, almost half of the genes are essential. So if you target half of the genes, you will kill the insect. But the critical issue is which part of the gene you will target. You may target the UTR, 5 or 3 UTR or CBS, but not the introns. You should definitely not choose intron sequences for targeting by double-stranded RNA or small RNAs, because the pre-mRNA, before splicing, the pre-mRNA in the nucleus cannot be targeted for degradation. Okay, so RNA degradation takes place in the cytoplasm and not in the nucleus. Thus, the pre-mRNAs cannot be targets for RNAi. And then another issue is to target essential genes, especially if you desire lethality, which is in most cases. And it's best if you choose non-conserved regions, in order to avoid the off-target possibility. Okay, of course, the non-conserved regions are less possible to generate some off-targets. Then another method practical method is how to apply this double-stranded RNAs. The initial attempts to apply double-stranded RNAs were with a conventional sprayer, and thus which is called a low-pressure spraying. In that case, the double-stranded RNA remains on the leaf surface when you apply it in the plants, and thus, because it remains on the leaf surface, it cannot enter in the plant cells and processed by dicers into small RNAs. But even like this, it's a suitable method for leaf-chewing insects because they just eat this double-stranded RNA. But of course, this low-pressure spray cannot be used to silence plant genes. In order to silence plant genes, you need to put some more pressure in the method, so we call it high-pressure spray. It's a pressure of approximately 5 to 8 bar. that is also, this pressure is also used in the conventional sprayers on the field, the tractors that have the sprayers, the sprayer machine in the back, spray any conventional chemicals, pesticide right now with average 5 bar. So this resembles the situation that's happening on the fields right now, the 5 bar situation. And with this method, the RNA, of course, does not remain on the leaf surface. It penetrates the mesophyll and reaches the plant cell. And as soon as it's in the plant cell, it will be processed by plant dicers into small RNAs, and it will also be transported through the phloem to the distant part of the plants. Thus, this method is suitable for to target plant genes, herbicides, for example, or viruses, viroids, and aphids, because we know that aphids suck the phloems up, so it's a suitable method for them. Another method is a petiole uptake, although this cannot be applied in the field. It's mostly laboratory method. But it's interesting because we know that with this method, when you apply RNA with this method, that you detach the leaf and from protruding petiole, you attach an epidorf containing your double-strand RNA, then the RNA is transported through the xylem and the apoplast, exclusively through the xylem and the apoplast. How do we know? Because we have seen it in the confocal here. We label the RNA, and then we show in the stem, the leaf, and the petiole that the double-strand RNA is transported exclusively through the xylem elements and remains in the leaf apoplast. And thus, because it cannot reach the plant cell, it will not be processed by dicers into small RNAs, and it will not be, that means that it's not a suitable method to target plant genes. But because the RNA will reach eventually the upper parts of the plants, including fruits, it's an optimal method to silence insects, the larvae that are feeding on the fruits, and they will take intact the double-strand RNA. And similarly, there is a track injection method. This method is, of course, because they are not all plants, potatoes or tomatoes. We also have some trees that we want to combat for pest management. And in the case of trees, a nice method is a trunk injection method. In that case, you just drill the trunk and you apply the RNA, as you see here in the picture. And the double-strand RNA is transported against, as in the previous case, through the xylem and the apoplast. And it reaches the upper part of the plants, including the fruits, where the insect or the larvae can suck the intact double-strand RNA and get killed, hopefully. Yeah, and actually, I would like to comment also this, that we also currently with Kalliopi Papadopoulou, we are doing some experiments on this and in several trees, woody plants, and it seems it's very promising method. And another method is the root drenching method. In that case, you apply the RNA on the soil. This is a very dirty method, and because the double-strand RNA in the soil gets immediately degraded after 2 days, it's degraded. If it's naked, if it's formulated, it's another issue. But if it's naked, it's degraded quite fast. But if you want to combat insects and fungi and nematodes, especially, then this is an option that you have to consider. Okay, so this is this cartoon. We see all the possible application of the double-strand RNA against several pests and pathogens, or against herbicides, plant genes, or against viruses, insects, and fungi. And I will go case by case. In the first group of applications, we have the double-strand RNA application against the plant gene. In that case, the double-strand RNA needs to be delivered in the plant cell, where it will be processed by plant dicer into small RNAs that will recognize a complementary plant mRNA for cleavage. Indeed, we have several examples in that group of applications: Arabidopsis, tobacco, tomato plants, also grapevine. They have their genes have been silenced with this method, and both endogenous and transgenes have been silenced with this method. The critical here is that not in all cases the silencing was systemic, meaning that some genes were locally silenced but not systemically silenced. The silencing was not spreading throughout the whole plant, which is of course desired when you apply a herbicide. So in order to explain a little bit further the notions of local systemic silencing, I will use some reporter plants. Here, for example, you see a plant, a wild-type plant, that you see green in the daylight. Most plants are green, of course. But in the UV, when you examine this plant in the UV, you see it red. Why? Because you have the chlorophyll autofluorescence. When you have a GFP-expressing plant, then you see the same plant, again, green, like the wild-type. But when you see it under the UV, you will see this plant as green, bright green. What is this bright green? It's the fluorescence of the GFP. Now, when you have the GFP expressing plant and you induce silencing in the leaf locally, in a leaf of this plant, what you will see under UV, you will see that the region that you applied the induction of RNAi is red. Why it's red? Because the GFP is silenced. And you see the remaining, the background chlorophyll autofluorescence. And the important part is that the silencing, in some cases, not always, silencing in plants spreads from the local tissue to the systemic tissue. Systemic is this part of the plant that you did not initially apply any RNA. Okay, so it's important to remember this cartoon to understand the next slides. So in order to understand or to examine which small RNA molecules are the most potent inducers of RNAi in plants, what we did, we used the GFP-expressing Nicotiana methaniana plant. This is a transgene that this plant has. And we targeted the GFP part of this plant with 21, 22, and 24 nucleotide small RNAs. These are chemically synthesized, in vitro synthesized small RNAs. Okay, so why these sizes? Because we know, as I said in the first slide, that the double-strand RNA is processed by 3 endonucleases into 21, 22, and 24. That's why we chose 21, 22, 24 here as well, to see what is the particular role of each one of these small RNAs. So when we sprayed these small RNAs independently into Nicotiana methaniana GFP-expressing plants, we saw that the 21 nucleotide small RNAs, these are all figures under UV, induced local silencing. You see here the red dots, suggesting GFP silencing, but no systemic silencing was observed. With the 22 nucleotide small RNAs, we saw some local silencing in the area in the sprayed leaf, and after 10 or 15 days, the silencing was spreading systemically to the upper parts of the plant. You see here, in 10 days, you see some spreading of silencing in the typical fishbone pattern, and after 15 days, the whole plant was silenced. Okay, so this is a nice example of systemic silencing, of spreading of systemic silencing. With the 24 nucleotide small RNAs, we didn't see no local and no systemic silencing. And this was not very unexpected. We just had to prove it because we knew that the double-strand RNA, I saw again the first cartoon, as I saw before, we know that the double-strand RNA is processed to 21 small RNAs that lead to degradation, but as we saw here, so these 21 small RNAs lead to local silencing, but perhaps their population was not enough in order to spread systemically into the rest part of the plant. The 22 small RNAs, we know that they recruit the RDR6 and they lead to the generation of secondary RNAs, thus amplifying silencing. And indeed, we saw this here, suggesting that the 22 small RNAs, or proving that the 22 small RNAs indeed lead to a self-amplification loop of silencing, and that's why we have systemic silencing. And with the 24 nucleotide small RNAs, we did not see anything, and it was not unexpected because we know that the 24s are involved in DNA methylation and not in RNA degradation. Okay, so this is the first take-home message, that the 22s are the most potent inducers for silencing. So if you want to use these small RNAs as herbicides. The next question we wanted to answer was whether all parts of any gene are equally susceptible to silencing. In order to address this, we designed 22 nucleotide small RNAs targeting the G, the 5 prime, the middle prime, or the 3 prime of the GFP. That's why I call it G, F, and P, okay? 5 prime, middle, 3 prime. And what we saw when we sprayed these small RNAs, we saw that local silencing was detected in all cases. So there was no difference whether one targets a 5 prime, the middle, or a 3 prime. However, when we followed these plans for the development of systemic silencing, we saw that only the small RNAs targeting the 5 prime showed systemic silencing, and this efficiency decreased the more downstream one went. For example, with the 3 prime small RNAs, no systemic silencing was observed. And we realized why this happened, because while we know that the 22 nucleotide small RNAs recruit RDR6, when they are in the 5 prime, they recruit the RDR6 through several enzymes like SGS3 or SD5, and they efficiently lead to the generation of secondary small RNAs. While when the small RNA, the 22 small RNAs is in the 5 prime, then it sterically hinders these enzymes and the RDR6 recruitment. Thus, even though it's 22 nucleotide small RNA, it cannot efficiently recruit RDR6, because remember, RDR6 is going always to the 3 prime part of the gene. Okay. So we concluded that the 22s are the best inducers of silencing. We concluded also that the 5 prime part is the best target region of silencing. And the final question was whether all genes are equally susceptible to silencing. And when I say all genes, in plants we have intronless and intron-containing genes. We don't have a third case, okay? Intronless and intron-containing. In order to see whether a 22 small RNA targeting the 5 prime is leading to equally efficient silencing in both intronless and intron-containing gene, we use the GFP expressing plants and GFP expressing plants, but where the GFP transgene contains introns. Okay. So what we did is when we sprayed the same 22 nucleotide small RNA, we saw local silencing in both cases. Okay, so far so good. But when we followed these plants for systemic silencing, we saw, as expected, that the intronless GFP plants showed systemic silencing, but the intron-containing GFP plants showed no systemic silencing at all. And we have an idea why this is happening, because the intron-containing genes, of course, they need to be processed by the spliceosome before the mature mRNA goes to the cytoplasm. And this association of the spliceosome is refractory to the RDR6. Okay, so any gene that contains introns cannot be processed by, its transcript cannot be later processed by RDR6. And without the RDR6 processing, we don't have amplification of silencing, and thus we do not have systemic silencing. And this fit with some data we had in the past, where we observed that the intron-containing genes, both intron-containing genes and intronless genes, are equally susceptible to local RNAi. But because the intron-containing genes cannot get processed by the RDR6, they are not susceptible to systemic RNAi. What does all this detail mean? This means that if I would like to develop a herbicide for in the future against any gene, I would make sure that this I would spray a 22 nucleotide small RNA targeting the 5 prime part of the intronless gene. Otherwise, I would fail. Okay, and because any herbicide needs to be, when it reaches the market, it needs to be formulated. The best formulation, in my opinion, right now is carbon dots. These are some nanoparticles that can be quite easily developed in the laboratory, where the RNA, the small RNAs, are protected against environmental degradation. and then they also facilitate the delivery of the small RNAs in the plant cell by endocytosis. So actually, that means that the application of small RNAs is working. It can reduce silencing, but if you use formulation with carbon dots, the silencing will be much more efficient, as has been shown in this very, very nice publication by Stefan Schwartz here. Okay, now the second group of application is when we target a virus, when we apply double-stranded RNA in the plants against the virus. As most of you know, most plant viruses are RNA viruses, meaning that they have an RNA genome. And in that case, the double-stranded RNA is applied in the plants. It needs, again, to be taken up by the plant cell, processed by plant dicers to small RNAs that will recognize the viral genome, the viral RNA, and cleave it. And indeed, there are several cases where this has been shown. The first one to demonstrate something like this was in Spain, Francisco Tenlado. He was a pioneer in that case because he was the first one to show that application of double-stranded RNA in plants makes plants resistant to viruses. And then, of course, several other publications followed in tobacco, in Cucumis sativus, in Zea mays, and other plants. As you see here, the plants, and the viruses, and the corresponding reference. But besides viruses, viruses are also very infectious agents. They are, unlike viruses, the viruses are not encapsulated, so they are naked RNAs. They are remnants of the ancient RNA world because they have a very small genome of 300 nucleotides, but they have no protein, so they are pure RNAs. But because they assume these extensive secondary structures, they are resistant to silencing. However, it has been shown by Ricardo Flores' lab that external application of double-stranded RNAs against both families of the virus, against the postviroid and adenoviroid, can also lead to their silencing. And when we speak about viruses or viroids, the formulation in that case for a commercial double-stranded RNA pesticides is the best formulation, in my opinion, is the BioClay, which is developed by Nina Meter Lab in Australia. The BioClay is a combination of double-stranded RNA and clay nanoseeds. What are these clay nanoseeds? These are these compounds that the RNA is encapsulated or attached to these compounds, and it's thus protected. They are protected, the RNAs, from the environmental degradation. Here data are quite convincing, and they show that in contrast to naked RNA, when the naked RNA is applied in plants against viruses and offers a protection window of 5 days, when the same RNAs are conjugated to this BioClay, they are much more stable in the environmental conditions, and they offer a protection window of 20 days. So it makes a big difference. It's 4 times more stable. Then the third group of applications is against fungi. In that case, the double-stranded RNA is applied again in crops, but it should not be taken up by the plant cells. It's not necessary. Actually, it's undesired to be taken up by the plant cells. Instead, the double-stranded RNA needs to be taken up by the fungal cell, of course, to be processed by the fungal dicer into small RNAs that will be loaded into fungal ago. In that case, it's not called ago. This protein is called QDE2, and the recognized fungal essential fungal mRNAs for cleavage. And indeed, this has been shown in several cases. One of the first to demonstrate this was Karl-Heinz Kogel in Germany, with Fusarium graminearum and barley plants. And then other reports followed in Triticum mastivum, tomato, vitis, chickpeas, against mainly botrytis, but also against other viruses. So all this, if one is interested, he can resort to these references. The interesting part here is that some insights coming from the Heiligen lab that demonstrated that the double-stranded RNAs from the plants are being transported to the fungal cells by exosomes. So this was really nice works that convincingly demonstrate this procedure. And now we know that any plant, any double-stranded RNA is taken up by the fungi through plant exosomes. And then the exosomes protect this double-stranded RNA, and then they release it in the fungal hyphae for RNAi. The important here is two things, actually. There are some limitations in this double-stranded RNA fungicide procedure. Not every fungus can be a target, because not every fungi encode the RNAi machinery. If you cannot silence, you cannot manage a fungus with RNAi if it does not encode the RNAi. And there are several fungi that do not encode the RNAi machinery. And when I say RNAi machinery, I mean mainly the AGO and the dicer genes, but also the RDRs. But the most important are the AGO and the dicer. So if your fungus does not contain the AGO or a dicer gene, then forget it. You cannot use double-stranded RNA against this fungi. Fortunately, there are not a lot. They are very exceptional. There are very few cases that do not encode the RNA machinery. I know only of 5 actually related to plant diseases. But it's important here to note that, for example, this fungus Usilagomydes does not contain the RNAi machinery, while its close relative Usilago-horti does. Meaning that it's not enough if a very close fungus to your fungus of interest encodes the RNA machinery. You have to ascertain it yourself that your fungus encodes the RNAi machinery by NGS. And the second issue is that even if your fungus doesn't encode the RNA machinery, you have to make sure that it's able to take up double-stranded RNA, because if it cannot take up exogenous double-stranded RNA, again, you will fail to kill this fungus. And in most cases, the fungi do take up double-stranded RNA from the environment. But there are some cases that where we have limited double-stranded RNA uptake, unfortunately, Phytophthora as well, and or even no double-stranded RNA uptake. These two fungi are completely resilient to double-stranded RNA uptake. No matter how much RNAi you apply them, they will simply refuse to take up anything. And it's important here, as in the previous case, that Coledotrichum gliosporides does not take up double-stranded RNA, while a very close relative does. So each case needs to be addressed specifically. And the final group of applications is the RNAi against insects. In that case, again, the double-stranded RNA should not be taken up by the plant cells, but by the insect cell, where it will be processed by dicer 2, the insect dicer 2, into small RNAs loaded in the insect AGO and target essential insect mRNAs for cleavage. This has been shown in several cases. One of the first showed was Hunter from USA, where he injected 6-meter lime trees with a double-stranded RNA, and he found that he recorded some significant protection against the Aphorina citri, and then some other cases also came up in different insects. But the most important here, the take-home message, is to remember that mostly coleopterans are susceptible to RNAi with this manner, not the others. As you see here, this is an unpublished data from Greg Watson from Bayer Monsanto. You can see here that the coleopterans are, the LC50 of coleopterans is quite low when you apply the double-stranded RNA, so they are dying quite nicely with with minimal amounts of double-stranded RNA, while the rest insects, Diptera, Hemiptera, especially Lepidoptera, no, they are very resilient. And why they are resilient? They are resilient because the double-stranded RNA is easily degraded in these insects. Either they spit nucleases on their food before they eat it, thus they degrade the RNA even before they take it inside their bodies, or there are some nucleases in their bodies, in the midgut and the hemolymph, that degrade this RNA. Thus, when if you are unlucky and you have to target one of these insects with double-stranded RNA, you have to somehow protect your double-stranded RNA. Unfortunately, this can't happen. There are methods to protect the double-stranded RNA against insects. There are several formulations. There are, for example, the liposomal particles, the hitosomal particles, peptide particles, and synthetic polymers, which each one has its own properties and is suitable for independent insects. There's not one rule for all. You cannot use, for example, dendrimers for all insects or hitosal for all insects. It depends on your insect. Okay, so it's you have to. It's a trial and error here. You have to try several formulations to see which is the best, most suitable for you. And fortunately, several of these formulations are commercially available. They can be bought, the dendrimers, for example, or the liposomes or the hitosal. But some other which are very efficient cannot be bought. For example, the bioclay that we spoke before. So this, you cannot buy this, and you cannot generate it quite easily in the lab. So this is very nice tool, but very hard to find. And now we're reaching the end. One of the first double-stranded pesticide on the market is a fact. It's coming from Greenlight Biosciences in US. And what is this commercial double-stranded pesticide? It is actually RNA, a naked RNA. We don't know whether there is a particular formulation of this RNA. There must be something, but we are not aware of what this is. So this double-stranded RNA targets the Colorado potato beetle proteasome subunit 5 gene, and it was approved just this year. And as you can see here, when you spray this RNA on the potato leaves, then the insect eats this double-stranded RNA. It takes up the RNA, and then its essential gene is silenced, and the insect gets killed. Here you can see this image from this company. Ledprona is actually the name, or Ledprona or Calantha. Calantha and Ledprona is the same thing, if you see it. Is the grams of active ingredient per hectares that offer this protection. You can see even with 0.3 grams per hectare, it offers significant protection against this beetle. Now, some issues that need to be taken into consideration when we apply double-stranded RNA pesticide in crops is not only to kill the target, which may be an insect or a fungus or a virus, but also what it does to the plant. Of course, the double-stranded RNA that we apply in the plants should not have a target in the plant. It should not be toxic. But this is not the only consideration that we need to be careful of, because when we apply the double-stranded RNA in the plant, then in the cytoplasm, it has the potential to be cleaved into 21 small RNAs to target an mRNA for cleavage, but it may also be transported to the nucleus, and then things change. Here there is no mRNA cleavage, but the double-stranded RNA may recognize DNA, especially if it has a minimal region of 30 base pair homology, and methylate this DNA. And when this, I will not describe how DNA is methylated, it's quite complicated. But this methylation of the DNA is an epigenetic modification that may be transgenerationally maintained in the next generation, even in the absence of double-stranded RNA, suggesting that if you induce with a double-stranded RNA herbicide epigenetic modification in the crops, when you apply it in year one, the next generation, even if the plants are not sprayed, they may have some carryover epigenetic modification. And this has not been sufficiently addressed. This is an important issue, because we like to have GMO-free plants, but we have to make sure that these GMO-free plants are also epigenetically modified free. Okay? And that in order to prove that the double-stranded RNA, sprayed double-stranded RNA can methylate homologous promoter or homologous DNA in plants, we have sprayed double-stranded RNA against a plant promoter, against a promoter in plants, and we show that, I will not bother you with details here because my time is running out, that the double-stranded RNA that was sprayed in this plant methylated the promoter of this plant, okay, after 10 days post-spraying. This we are certain by bisulfite sequencing. And so this issue of epigenetic modifications and other issues are being addressed right now in the project Ration, where Dimitrios Karpouzas is a coordinator. And so we try to actually assess the risk of low-risk pesticides, which are not only plant extracts and biochemicals, microbial pesticides, and double-stranded RNAs, which is where I'm involved. And we are trying to address issues that have not been properly addressed or insufficiently addressed. And so what we try to do is to identify first the hazard of double-stranded RNA pesticides, how to do this by developing bioinformatic tools to identify possible non-target organisms, because you may target, for example, the Colorado potato beetle, but you have to make sure that the double-stranded RNA has no homology to a bee, the pollinators, or some other non-target organisms. Then we also address the issue of genetic modification in crops and in insects, and also other issues like the saturation of the RNA machinery or the immune system stimulation. And the next step is to characterize the hazard by toxicity and ecotoxicity tests, and finally to assess the exposure of double-stranded RNA pesticides by measuring the environmental fate of double-stranded RNA in soil, in water, and crops. And importantly, we investigated, our postdoc Venetia Koridou investigated the translocation of the double-stranded RNA in crops, and also in pollen. It's important to see how the RNA in crops is moving and whether it reaches the pollen or not. And of course, in any risk assessment scheme, the formulation issue has to be taken into consideration. And that's why all the analysis that we do here, as I spoke before, include the naked and formulated RNA. And in this Ration project, there are several partners throughout from Europe and also from the non-Europe partners, like here Weissman, Genolution. Genolution is a Korean company that provides us with a double-stranded RNA. So we have the University of Thessaly, which is coordinating this project. We have companies from universities from Germany, from Wageningen, Belgium, Spain, Austria, France. So it's really a nice collaboration here. And with this, I would like to finish. These are the people that are involved in Ration double-stranded RNA pesticides. Not all the people that I'm working with. There are several people working with this. So there's Venetia Koridou, which is a postdoc that is mostly doing the experiments, and also Kalliopi Papadopoulou and Dimitrios Karpouzas as the coordinators of this project. So thank you very much.
Okay, thank you, Athanasios, for this very exciting talk on the possibilities of double-stranded RNA. You know, so some very nice example of applications in different fields. So we have time for a couple of questions. I see Mohammed. No. Does somebody ask questions?
Hi, Stefan. I have a question.
Yeah, okay, go ahead.
Hello, Athanasios or
Hello,
Thanasis. Nice to meet you.